Hi, I'm Stacey Blaine from Concarlo, and we are a precision oncology company generating a brand new class of products in order to transform the way we treat drug-resistant cancer. And all of our efforts are centered on a new target, P27, which has the unique property of controlling drivers of cancer progression and drivers of drug resistance. And we're looking to raise $3 million now in a seed round. Well, the oncology field is filled with many different targeted therapies. Targeting CDK4 has been a holy grail in the cancer space for several decades because inhibiting it's essentially downstream of all the other oncogenic drivers. And inhibiting CDK4 should, in theory, be able to deal with the downstream resistance that we see in the presence of all of these RAS MAP kinase targeting drugs. The CDK4 inhibitors came to market in 2015, and these are great drugs, but we now know that there's room for improvement. These drugs essentially increase the remission window of patients taking them twofold, but in most cases, overall survival is essentially unchanged. And the issue is that many patients are initially resistant to the CDK4 inhibitors, and those that initially respond eventually become resistant. So to really improve outcomes in metastatic breast cancer, we have to develop drugs to deal with this specific resistance. We now understand that targeting CDK4 is not enough, that even when CDK4 is inhibited, CDK4's twin, CDK2, is able to rise up and do CDK4's job. So to create a fantastic drug, you have to inhibit both CDK4 and CDK2. Drugging CDK2 has really stumped our field for several decades. And so at Concarlo, we took a different approach. We target the one common denominator between CDK4 and CDK2, and that's the protein P27. And IPY is Concarlo's P27 specific inhibitor that inhibits CDK4 and CDK2. And while we're initially going after metastatic breast cancer, our addressable market is much larger because P27 targets the RAS MAP kinase pathway, which regulates drug resistance in many other cancers, such as listed on this slide. And we plan to move into those markets as well. I'm only going to show one piece of data, and this is in a breast cancer palbociclib resistant mouse model, and we're plotting overall survival. And so when we treat these animals with palbociclib, they're all dead by day 20. But when we treat them with IPY, Concarlo's drug, or a combination of IPY plus palbociclib, we can keep the mice alive for over 65 or over 100 days. And so we have taken a biologic approach at Concarlo based on the natural P27 inhibitor that is lost in cancer cells. And so we basically are using nature's high throughput screen that they perform to identify the best P27 inhibitor, and we're delivering it via liposomal technology. And we plan to be first in human in 2022. We have another approach, which is a more pharma-friendly small molecule approach, again, based on the fact that we're the mechanistic experts in this area, and we have designed a proprietary screen, and combined with computer modeling, we hope to identify some small molecule hits that will mimic the peptide gold standard. Another differentiating fact for Concarlo is that we, even though we're relatively early, we have a seasoned mix of business and science experts. So the company is run by Lisa Marie Casey and Jason Mraz with their expertise in strategic and financial markets. I'm a molecular cellular biologist, having trained at Princeton, Columbia, and Sloan Kettering, and I've been thinking about P27 and CDK4 for over 20 years since my postdoc with Juan Masquet. Dominique Berdone heads up therapeutic development, having worked with some of pharma's biggest names. Our scientific advisory board is world-class. Gary, Neil, and Jeff, really, they are leaders in this field running clinical trials at their respective institutions, and they're instrumental in our journey. Our IP strategy focuses both on composition of matter and mechanism of action. It is led by Lisa Hale at DLA Piper. And we've done everything to date on a little under $4 million raise. We have been successful in raised SBIR funding. We have been awarded a contract from the NIH Nanoparticle Characterization Lab, and we're ready now to raise our $3 million seed round, which will finish our biologic manufacturing and get ready for the clinic, as well as finish the diagnostic work so that we can out-license that product and actually finish our first small molecule screen so that we can then fundraise around those hits. So the advantages of targeting P27 are pretty clear. Toxicity is low, specificity is high. We hit 2 targets for the price of 1, and we hope that you've liked what you've heard and you'd like to hear more about Concarlo. Thank you.